Well, hello everybody. Thanks for joining us for, I think, what is the first common read of the Purdue Ag Alumni Association. We appreciate y'all taking the time to join in, and I'm especially delighted that we've picked my book to talk about for this first time, Unnaturally Delicious. What I thought I'd do here at the beginning is just give you a little bit of insight as to why I decided to write this book and give you hopefully you've read it by now, so spoiler alert, I might hit a few high points on the first four chapters of the book. And I've also got a guest, as you can see here with me, Steve Lindeman. He's a professor in food science. So after I talk for a few minutes, Steve and I will have a little chat, and hopefully this will be a great opportunity for you to learn a little bit about Steve's research and some of the really interesting things that are going on here at Purdue. And then we'll also give you a chance to ask some questions of me and to Steve. And so as we're talking, feel free to type in there to Facebook if you have some questions you'd like us to answer. We may not be able to get to them all, but we'll do the best we can and at least try to pick the most interesting ones. So the book, I decided to write this book in large part because, frankly, I was a little frustrated with a lot of what we read in the media about food and agriculture in a lot of best-selling books and in the newspapers. There's a lot of pessimism about food and agriculture, and often a lot of the solutions people propose to dealing with our big food and agricultural problems are often a kind of return to nature sort of solution. And there's nothing wrong with that. We have a big diverse food supply. But my view on things is that if we really want to make substantial progress in solving our big food and agricultural problems, whether it's climate change or obesity or soil and nitrogen runoff, we're going to need science. And we're going to need innovation. It should be one of the tools that's on the table. And I had the great pleasure of working in land-grant universities, seeing people around me doing really fun and interesting things. And I think people would love to hear the stories of people on the innovation side of this. And so, I think one thing I thought I'd do here at the beginning is just, tell you that I think despite a lot of this pessimism about food and ag, if you look at the data, there's really been remarkable changes in food and agriculture over the last 50 to 100 years. So just to give you one example, this is a thought experiment that I think is particularly useful. Let's take dairy. Let's say that we as a country wanted to consume the same amount of cheese and milk and yogurt that we actually consume this year, but we wanted to do that using 1950s technology. And by 1950s technology, I basically mean how much milk per cow. How many more dairy cows would we need? And the answer is about 30 million more dairy cows. Now, mind you, we only have 9 million, so we need to basically triple the number of dairy cows that we have out there if we want to, enjoy the actual amount of milk and dairy that we consume this year, but we're doing it using old technologies. So sometimes I think it's, people get lost in those numbers, but it's really an enormous achievement. It means that we need this 30 million fewer dairy cows that are needing food to eat, that are emitting waste potentially that could get into our water, that are emitting carbon emissions. So I think that's really a great story, but in the numbers, sometimes it's often easy to forget that there were real life people working on all those innovations. People working on better feeds, people working on better housing conditions, people that were innovating new genetics, that were studying that side of it. And so that's really, I think, a large motivation for this book is to try to take those numbers that we can all see, that our eyes kind of glaze over a little bit about, and then look at the people that are behind those numbers. So the first chapter was an introduction. The second chapter was a chapter about animal welfare, and talked there about mainly chicken housing, because that's one that's been the subject of a lot of policy controversy. And again, trying to put forward this idea that a lot of people, I think, in their mind think, if we want to make chickens' lives better, let's just open up the cages and let them roam free. One of the challenges with that, of course, is if chickens are outside, there's weather and dogs and wolves and other problems. So we tend to bring them indoors. And so we need ultimately, I think we need some science. What are the things that we're trying to do to improve animals' lives? How do we know it's actually working? So I talked about some innovative housing systems there, and towards the end of that chapter, talked a little bit about some potential innovative economic systems that might be able to improve animal welfare. And I'll just put a little plug in here. I was fortunate enough to give a talk at a Purdue TED Talk series here on campus last spring, so hopefully we can throw up a link to that so that you can see a little bit more of my discussion there. The second chapter of the book talked about 3D printed food and robotic cooks that could enter our kitchen one day. We're not quite at the Star Trek food replicator where we just push a button and have our meal, but we're making small steps that are leading us in that direction, and I think those are really exciting. I'm particularly excited about the idea maybe of just being able to come home after a hard day's work and enter a recipe into a computer and then have some robotic arms go to work and do the cooking for me. It doesn't mean I can't do it when we want to, but those are some exciting ideas that I think were kind of fun to talk about. And then the fourth chapter, and this kind of leads into a little bit of our discussion here with Steve, was about this area of research called synthetic biology. And some of this is related to using genetics to genetically engineer bacteria or yeast to do interesting things. So for example, if you're a diabetic or you know anyone who's a diabetic, that insulin you took, or they took, was almost certainly created by a genetically engineered yeast. We put the genes into the yeast to produce the insulin that you now take, and that might sound a little scary, but you have to realize that before all that insulin came from the pancreas of pigs. So you got to pick your poison, I guess, on that one. Or, for example, cheese. If you've eaten cheese in the last few weeks or months, almost certainly one of the ingredients used to make that cheese is an enzyme called rennet that was almost certainly created through biotechnology. So those are some of the applications. I interviewed several students that were involved in this competition called iGEM, Internationally Genetically Engineered Machine Competitions, and they're using synthetic biology to do interesting things. I talked about a student group that created a sensor for rancid olive oil or bad tasting olive oil, and then a team that thought a little bit about how to use genetically engineered E. coli to maybe improve our gut microbiome to consume the fat before we do and turn it into something helpful. And that's really where I kind of ran into Steve. And so Steve works, as I mentioned, in food science. And so maybe rather than me trying to explain to the audience what you do, I'll just ask. So first, Steve, why don't you start off and just tell us who you are, how you got here to Purdue, and then maybe what you do.
Hi, I'm Steve Lindeman. I grew up in Northwest Indiana. I graduated from Munster High School, go Mustangs. Did my bachelor's work here at Purdue in microbiology and molecular biology. After that, went to the University of Iowa and then studied some pathogenic bacteria interactions with humans, and learned microbial ecology after that at Pacific Northwest National Lab, and then got to bring all of those things kind of united into one whole here back at Purdue.
Yeah, well, great. Well, it's, I know in talking to people around here, they're happy to have you back. So
it's good to be back.
Yeah, well, good. So, what is your role here at Purdue? What is it that you do your research on and that you're focused on?
So in general, I study diet microbiota interactions, so that we're interested in what happens to food after you eat it, and how it's processed by microbiota that live inside you, and what that influence might be on your health.
Yeah.
So specifically, mostly we work on fiber, dietary fiber, which everybody reads on the side of the box and knows that they should get more of. We basically are interested in how fiber structures are influencing the gut microbiota, and what the microbes do with the fibers they consume that might be beneficial for us, or maybe not so beneficial, but we think mostly beneficial for fibers.
Yeah, I think most of us kind of know that there's a lot of bacteria living in us, but well, at least if you're like me, I don't like to think about it very much. But it seems that this, our microbiome, I guess they call it, gut microbes, seems like it's really important for health. So what do we know about our gut microbes and what they do for us, or how important that is to our health?
One thing to think about, so first, humans are pretty efficient at eating what they eat, right? So with respect to the small intestine is really good at removing nutrients, but lots of what we eat still ends up in the colon, including dietary fibers, some undigested proteins. And you have to think about the fact that pretty much anything we get in that space, the microbes are eating first. So some of that is they're making things available to us. With respect to fibers, we think that about 10% of human energy balances could come from fermentation of fibers that you can't break down, but the microbes that you eat can. And so many of the metabolic products of what they make are the preferred food sources for our epithelial cells inside of our gut. So that's, we're adapted to that kind of production from them. They also produce vitamins we can't produce. Vitamin K would be the best example there. If you like to clot when you cut your hand or whatever, vitamin K is really important. So
I like to clot. So
okay,
that's
good.
Good to hear.
That's good. keep the microbes around then. And finally, they also mediate a lot of our drug interactions. So they can sometimes metabolize drugs before they get to us, either making them more effective, or sometimes a problem, sometimes making them less effective. And so xenometabolism, what we consider the metabolism of compounds that are not normally in the body, is an important subject by the gut microbiota, understanding how they might be modulating what happens to the medicines we take.
Interesting. So I've even read, studies, for example, I think these are mainly mice studies, but where they'll take bacteria from a skinny mouse, put it in the gut of a fat mouse, and that fat mouse will get skinny. And that's kind of amazing to me in terms of what impact our microbes in our body can have. But so I want to kind of get to the nuts and bolts of this. So we go in the grocery store. There's a lot of probiotics for sale. Whether it's yogurt or even pills we can take online. What do you think of those? Should I do those? Are those helpful? What do we know?
So I think most of our research is suggesting that probiotics are really very specific thing, and it's very difficult to know whether you've got the right organism in the pill. I mean, the organisms we put in probiotic supplements tend to be the ones that behave well for us. They tend to be the ones that make, for example, good yogurt. But whether they are involved in mediating health is frequently really specific to the strain or even the genome of the organism in question. So what that could mean is you could get the right species of bacterium and not have the exact right organism that would be beneficial, and the other one may be completely washed out or not help you in any way. And so we're just getting to the point, I think, where we're starting to get some traction in that space. I would hold off on the probiotics for the moment. They're probably not going to hurt you, but may not help you either.
Yeah, so I think you had mentioned to me this analogy of planting trees or something. How is that like this, taking a probiotic?
Oh yeah, so I mean, the point is that the gut is an ecosystem. And so because it's an ecosystem, you find certain species there that are adapted to that space, and kind of everybody's ecosystem is a little bit different. I think the analogy you're talking about is there's a reason there's not a bunch of tall oak trees in the middle of North Dakota. The environment doesn't support tall oak trees. You can plant oak trees all you want, put those acorns in the ground every day. You're still not going to get an oak forest in North Dakota unless you water it, unless you continue to nurture that ecosystem. Same thing why there's no giraffes out there either, right? They're not adapted for that space. So if you pick an organism that's not adapted for the space, well, it'd be the same thing. You can plant or seed those species, but they're just not going to colonize. They're not competitive enough to compete with the rest of the organisms out there and establish a toehold.
Yeah, I mean, I assume that makes the science really complicated because, as you said, it's an ecosystem. It's not, just a single thing. And it sounds like maybe, you might have North Dakota in your ecosystem, and I might be Texas in mine, or something along those lines.
Yeah, and that's where the hard part is. That ecosystem is shaped a lot by what we eat, and we all have different diets. And so understanding those diet interactions with the microbiome is really important to try to figure out what parts of these things are idiosyncratic, they're just things that are individual to you or me, and what part of these things are generalizable, things that we can use to engineer foods or engineer organisms to accomplish purposes that we want them to do in the context of many different people's guts.
Right. So you had mentioned a word to me that I hadn't heard before. So there's a probiotic, but you also said there's something called a prebiotic. What is that all about?
So a probiotic would be an organism, again, that you would be delivering to the gut. A prebiotic is a food source for the microbes that would be beneficial, we hope, inside the gut. So we would be adding, mostly when we talk about prebiotics, we're talking about carbohydrates, and specific carbohydrates that are designed to be more targeted towards the organisms we think are more beneficial. Or at least the biology we think that's more beneficial, more fermentation and better production of acids that we think are more healthy.
Yeah, that was really an eye-opener to me. I think about feeding myself. I never thought about feeding the microbes inside me certain things that they might want to eat too.
And so the reality of that circumstance is if we're not feeding our microbes well, they've got to eat too. They'll start to eat us, we think. So, our epithelia of all of, our intestinal tract is lined with mucus. And it's a glycoprotein. It's a mixture of sugar and protein. And if we don't feed them the dietary fiber or those kinds of carbohydrates that they like, well, they don't have anything to eat. They'll start to eat our glycoproteins. They'll start to take off the sugars, and it'll reduce the barrier that we have between us and them. That's not so good.
Yeah. So, I'll bother you with one more kind of practical question. There's a lot of dietary advice that goes out there. So, the probiotics was one of them, but we see things, sometimes on Facebook or in the grocery store, like you should drink cider vinegar. It's good for your, gut bacteria. Or kombucha is a fermented beverage. That's good for your, gut bacteria. What do you think about those things?
So there's some argument that the metabolic products in kombucha or in vinegar is acetic acid, which is normally produced in our guts and we think is pretty important in terms of maintaining health. And so that's a good thing. So maybe eating some of that is valuable. With respect to the specific organisms, we fall into the same problem of there's just not enough really strong data that show these organisms or this preparation of organisms is really beneficial. And so a lot of the health claims and the wisdom that we see passed around, we from a scientific aspect, we kind of have to shrug at. It's really, we don't really know, and there's lots more research needed.
Yeah, well, it's good we have people like you working on that. But
good job security.
Yeah, right. Lots of unanswered questions. The challenge is keeping up with all the new innovations.
Absolutely.
As a food scientist and somebody who spends his life studying these products that go into our foods and understanding how our body digests them, do you find yourself sometimes frustrated with a lot of these claims that you see on the supermarket, or do you have thoughts about how we could think about those better, or what do you think when you see those?
It's not so much the claims on packages I worry about, because those are very tightly regulated most of the time. Except when you're in the supplement aisle, you're in the Wild West, so anything goes there. But with respect to the foods, those are very tightly regulated. It's the things that bother me or concern me are kind of the expectations being put on gut microbiome research and understanding, using it to improve health by the consumer base are well ahead of, I think, where we are with the science. And so, I'm afraid that people have expectations that are just of things being curative that we just have no or very little data about right now. So we're just learning these things. So, if I could encourage anything of people, it's be patient, we'll get there, but maybe hearing about it, third hand from somebody else is probably not going to be your source of a panacea. And, everybody's looking for a nutritional panacea. I don't think those probably exist.
Yeah. Unfortunately, I think that's right. We want silver bullets, and the world is not that kind, unfortunately. So, despite, I think you're warning a right warning to folks to say, don't get too over optimistic here. At the same time, I'm going to try to make you be a little over optimistic here and say, you are a young guy. You've got lots of years in front of you. What do you hope to accomplish in your career with sort of your research? What's kind of the holy grail? If you could find something, maybe, Steve Lindeman wins the Nobel Prize one day. What would that be for? Like, what do you hope to really accomplish?
Yeah, I don't think that is a likely outcome, but thank you for the vote of confidence.
I said we're being overly optimistic.
Okay.
Here. Yeah.
So I think the contribution I'd like to make is understanding what's generalizable about dietary fiber carbohydrates in terms of how they interact with the gut microbiome, which structures are specific for which species, which kind of metabolic functions come out of certain structures or different species. So we get these interactions all figured out in ways that we know what works across many different people. And so we can start to predict how the gut microbiome is going to respond to diets or to specific kinds of carbohydrates. And we can use these things as tools to control it. So once we figure out what we want, and I don't think we figured out what a healthy gut looks like yet, which is another problem in microbiome research. Once we figure that out, we will have the tools available to move gut microbiomes to healthy space from unhealthy space. So we'll know how to engineer the system and control it and manage that ecosystem. It's like the analogy would be, a gut-controlled burn, right? We want to be able to manage an ecosystem in a way that is less, it's not harmful to the ecosystem, but we get the benefit of ecosystem services or the production from that ecosystem that we like and enjoy. So, we want our guts to keep us healthy. We need to be able to manage it in order for that to happen.
Yeah. So it sounds to me like, and correct me if I'm wrong, you're saying you really want to help develop a theory of why our microbes in our gut work the way they do. And then maybe if we have that, then when a new product comes along, and we want to know what does this do, we'll have a much better idea of doing that rather than having to run a little experiment every time.
Absolutely. I mean, the reality of the circumstance is that carbohydrates are so structurally diverse that it's impossible to test every combination.
Yeah.
We have to have a theory that describes what aspects of carbohydrates are important. In this case, I'm thinking about fiber carbohydrates or carbohydrates we don't consume, but microbes do. What aspects of those carbohydrates are important for governing the gut microbiome's structure, for governing its function, for nourishing certain species or not. And those have to be as generalizable as possible, and we need to know what the limitations are to their generalizability. So we can actually talk to people, real people who have real diets, and say, maybe you should consider this supplement or that supplement, rather than, well, they're all just kind of the same. Go eat your fiber. Well, good advice. It's good advice to the people get their fiber, but maybe there are certain fibers that are more active than others or more targeted than others, or maybe there are certain things, organisms we want to encourage or discourage more than others. We just don't know yet.
So right now when I pick up a package of food at the grocery store and it's got the nutrition facts panel, it tells me the number of fibers, or number of grams of fiber, for example. But what you're saying perhaps is that not all those fibers are created equally in terms of how our body processes them?
It's still early evidence, but our evidence suggests that very fine differences in fiber structure make big differences in the gut microbiome. And so it's maybe even as simple as hard red wheats versus soft red wheat fibers, or big particle sizes of wheat bran or small particle sizes of wheat bran, or white versus red sorghum. Different responses from the microbiome. And none of that information is anywhere on your package. So at this point, how would a consumer even make a decision based on those parameters? But we're hoping to get towards informing that space, and kind of both from a technology side, but also from a policy side. We're going to have to change some of the way we think about fibers and foods.
Yeah.
And so, we need science to do that.
Yeah. So the message is now eat more fiber, but the message in the future may be eat this specific kind of fiber.
That's exactly the hope.
Yeah. Well, that would be useful. I'm going to put you on the spot. I don't. And see if you actually read a part of the book, but I thought I'd see what.
It's quiz time.
Yeah,
quiz time.
That's right. What you thought about some of the kind of synthetic biology applications that I talked about. So there was this team of the students from that developed this E. coli to eat some of the fats in our food. I could see some people being a little fearful of what some unintended consequences. But what do you think about that?
Sure. So there's certainly. The tool outstrips our understanding of the ecosystem. Again, you wouldn't want to. There are some organisms you don't want to engineer and set loose in the wild, which is essentially what you would be doing in that case. But I love the approach. The idea that you can take a nutrient that there's an undesirable nutrient and turn it into something that is a desirable nutrient using microbial activity, I think is spot on. I really like the biosensor approaches that you described. I think even some of those could be useful in the context of the gut, like swallow a pill full of a biosensor organism. Yeah, it never comes out into your gut, so it's not at risk of causing trouble in your world, but it can sense whether it saw specific nutrients and the concentrations of those nutrients that it saw, or whether it saw oxygen, or whether it saw some other aspect, good or bad, in the behavior of the system, and you can just easily read it out. Can you imagine what that would be?
Yeah, that's awesome. That's a really interesting thought. Well, I've been bothering you with a lot of questions, but I thought I'd go online and see if some folks typed in some questions. So it's always a little nerve-wracking. You never know what people are going to say, but let's see here if we have some questions online. So some of these might be for me about some of the other chapters, and some might be for you, but I see one that's addressing another one of the chapters in the book, so maybe I'll take that one.
Go for it.
We'll get back to your area of expertise, although you can feel free to chime in and help me out too. So one of you asked about the animal welfare chapter that I wrote about, and I talked about these things. I called them animal well-being units, and somebody said they thought this was interesting, but how could these be combined with other approaches? What other approaches might we consider? So, my training is as an economist, so I like to try to incentivize behaviors that we desire and get people to think about the cost. Actually, before we came online to here, Steve and I were talking about, we'd all like to have, kind of free-range animals that have lovely, lives, but we also want affordable eggs, and we want inexpensive meat. We want our cake and eat it too, and I think that's one of our challenges in these areas is we got to think about the trade-offs. and about the costs of the policies that we think about. So, this animal well-being unit approach that I talked about, this is actually one that's being used in environmental applications. So markets for carbon, for example, is really where I sort of took the idea for these animal well-being units, that we want to, provide a monetary incentive for folks to think about these things. And I think we can think about some other interesting market approaches for water use, even for soil runoff and some of those things. This is a question, Steve, I think falls squarely in your area of expertise. I wrote a little bit about in these synthetic biology applications that often people use E. coli as one of the bugs, so to speak, to convey some of these things. Are there any dangers in using this E. coli in synthetic biology applications?
I don't really worry about E. coli as an organism. E. coli, the lab varieties of E. coli, even the commensal ones that we carry around, almost all of us carry around, at least some, they're not harmful to us. They don't have the genes to be dangerous. However, so I would also add that E. coli is the best understood organism on the planet, probably, right? So even though it is the best understood organism on the planet, we still probably don't know what 30-40% of its genes are doing. So, that gives you an upper limit for our knowledge on that space. So, it's really well understood, not a risky organism. Now, the thing we do have to worry about is what kind of genetic engineering we're doing. So we just want to make sure that, this is an example of the probiotic we talked about. It's like you can engineer organisms that maybe are more competitive in certain spaces than organisms you actually want to keep around, and they might outcompete. So we have to worry about recombinant DNA, and we just have to make sure it's done safely. It's definitely a technology that we use every day, but this is why institutions such as Purdue have institutional biosafety committees to make sure that we're not doing things that are dangerous.
Yeah. There's an interesting question that came in here, and this we both may be out of our comfort zones, but I think it's an interesting one too. And it's moving into the plant space, but we talk about our gut microbiome, but there's bacteria in our soil too, and there are, I know there are researchers working on sort of the not the gut microbe, but the microbiology of the soil, and talking about these applications where instead of injecting a fertilizer or pesticide, that you're injecting microbes in the soil. Do you know anything about that, or what do you think about some of those approaches?
Actually, I think this is going to be where a lot of soil management is going to be a lot of where we're going to see gains in productivity in agriculture. Microbes are known to be really important for growth promotion of plants. They cycle carbon, phosphorus, and nitrogen in ways that are advantageous. Some of them are, really important organisms that are symbiotic with plants, like we think about rhizobia and interactions with soybean as being kind of the type case or one type case of that. But there are many other interactions with between microbes and plants that are really helpful for growing plants. In fact, if you grow like model plants like Arabidopsis in sterile soils, they don't grow nearly as well as if you give them unsterilized soils. So there's something really important going on there. I think over time we're going to learn to optimize it. And the other option is you can use microbes sometimes for pathogen control. Biocontrol is not a new idea. People have been using biocontrol in microbial ecology of soils for 100 years.
Right.
To try to suppress pathogens and to keep soils healthy for crops that we're interested in. But I think that as we get more traction around that and more information on the interactions going on in soils, I think we have a fair bit chance of improving both yield and then also disease resistance using microbe-mediated approaches.
Yeah. So this probably is a big question, but it's maybe one we can kind of wrap up on. But this is a question about what it will take to encourage innovation and entrepreneurship and science in food and agriculture. So, you can take that on as a broad question, but maybe also just to say, what does it take to run your lab, your program? How many people work with you, and what does it take for, you to kind of do what you do?
Yep. All told, we're about 12 people right now in the lab, various different levels. So we have all the way from postdocs and visiting scholars down to undergrads, and so that requires substantial investment. The thing that I think is first and foremost is the public needs to express that they care and support to people who actually make decisions. I mean, the reality of the circumstance is everybody wants to know what they should eat, but as a society, we're not very good at funding those answers to those questions. And so we get a lot of confusing advice.
Yeah.
So what my suggestion would be is let people who represent you in government know that you care. so that folks like who are doing the research can be supported in that.
Yeah, I think that's a great point. I was asking these questions earlier about, should I take a probiotic? Should I drink this kombucha? And you kind of say, well, we don't really know what the science says on those. But if people really want to know, they need to fund the research to make it happen. And I think if you look at public funding for ag research, it's been on the decline, at least in real terms, in recent years. And so I think that's a great point. So I think with that, folks, we're going to wrap it up, but I'd encourage you to keep following along. Our next Facebook Live event is November the 2nd at noon Eastern Time. I believe we're going to have Bruce Erickson talking to us a little bit. He's in the agronomy department. I think we're going to be talking about some precision ag issues. The next chapters are 5 through 8. Feel free to reach out, send me questions, and thanks for taking the time. And Steve, thank you for being here.
Thanks for having me.